Let's talk about meta-analysis. As Susan said, it's a methodology that has become increasingly important. This afternoon, in fact, so I went online, got onto PubMed, and said, so let's put in epidemiology and just meta-analysis. You know how you can do that, just to sort of find the meta-analyses. So in 2013, you get 3,200 hits. In 2012, 2,700. So this is a big deal. If you go back in time, back to when I was a youngster in this, I'm now 30-some years into this, it was non-existent. I mean, meta-analysis as a methodology is really fairly new as a methodology. Probably the mid-1980s is when it sort of hit the fan here and we started talking about it. There was a discussion in the epidemiologic literature in the 1990s. There was this very famous paper called Meta-Analysis, Schmeta-Analysis, written by Dr. Cohen, who said that, not the Cohen you know, who said, no, this is a bad idea. Well, that didn't turn out so well for him. I mean, it's a very popular methodology. And so let's talk a little bit about it and some of the strengths and limitations and how it works. And I'm going to focus primarily on meta-analysis of observational studies. I'm an epidemiologist, and though I believe very strongly in randomized clinical trials, the issues are a little bit different for those. So we'll talk about observational studies. Now, you've seen these pictures. So you're all sort of comfortable with what these are. This is actually not a carbohydrates. This is actually a colorectal and red meat meta-analysis. But the reason I like to show this is that if you think back, let's pretend we're not in 2015, we're in 1982, before meta-analysis, and what was your task at that point in time faced with exactly this evidence? Somebody had to look at that evidence and say, okay, so, and I wish I could walk over there, but they told me I'm not allowed to because then I can't record it. So you've got all of these various studies. These are a whole variety of cohort studies of epidemiology, each one of which has a relative risk measure and a confidence interval around it. And then someone were to ask you, so what's the magnitude of the relative risk for all of those studies overall? And I can remember my advisor, epidemiologic advisor, Dr. Tyroller, and he had his glasses down like this, and he'd look at you, look at the paper, and he'd go, I think that's about a 1.2. Who could argue with Dr. Tyroller? So prior to the 1980s, we had no way of doing what now seems like such an obvious thing to do, which is to find a way, and you see the result down here if you can. It's not important that you see the actual number, but you actually have a way now to average those results across all those studies. This is a big deal. I mean, it's an important thing that we can do, and not only can you provide that average, and we'll talk a little bit about how that works, but it's also a more precise average, a precise number than you had before. So the confidence limits around all of those numbers have now shrunk, so that instead of some of those wide ones, we now have a relative risk down here of 1.1 with a 95% confidence interval between 1.03 and 1.18, which is a pretty tight, precise estimate of the relative risk. Now, there's something else there about heterogeneity, and we'll get to that. But that's what meta-analysis is all about, in a sense. But there's a bigger thing about meta-analysis, a bigger important issue about meta-analysis, and that's how it fits into these methods for synthesizing evidence. We're all faced today with this sort of larger problem, which is I've got evidence and I need to make something out of it. I need to come up with some interpretation, a causal interpretation or not. And in that context, meta-analysis is just one, but not the only, synthetic method. By synthetic, I mean research synthesis, as in putting evidence together. It's an important method, but it's limited because it's not by itself, it does not provide you with enough information, for example, to make a claim of causation. not when you're talking about epidemiologic evidence. So these are actually the methods that you use to interpret evidence. There's this general scientific method, which is the method that we all know about and no one ever writes about. I mean, when was the last time you read a paper and the first part of the method said, oh, by the way, we're using the scientific method and this is what we mean by that? It's there, but we don't write about it. But that's the overarching method, the method of predictions and observations and tests of hypotheses and trying to find the best explanation from those alternatives. That is the method that we're all using. Now, what we tend to like to talk about are the study design and statistical methods. Those are the methods that we're all comfortable with. Those are the sort of study design and study methods. And then we have these synthetic methods. And these are the methods we use to combine that information. The systematic narrative review, meta-analysis and pooled analysis, and the criteria-based methods. And meta-analysis, as I mentioned, fits in there. And it does at least 3 things for us in that process of evaluating evidence. As we talked about before, it gives you a weighted average of the observed effect measure. So if I've got a variety of studies here, and each of them has their own estimate of the risk for whatever that is, whether it's carbohydrates and type 2 diabetes or whatever, it's going to give me a weighted average of that. And that weighted average, the weighting, is going to be a function of the size of the studies. Very fundamental, but very important idea. The bigger the study, the more the weight. The second thing it does, and some folks say that this is a more important feature than the first feature, although it doesn't really matter whether you think it's more important or not, it's an important feature, is the assessment of heterogeneity. That is, and we're going to talk more about heterogeneity in a little bit, about what that means. It is about the differences in those studies. Because everyone knows that no study is exactly the same as some other study, and some studies are more different than others. Very simply put, you don't put studies together that are too different. Let me give you a great example. How many toxicologists are there in the room? Anyone admit to that? Oh, that's good. When was the last time you saw a meta-analysis that combined animal studies and human studies? You would say, well, of course not. I mean, that's silly. I mean, why would you ever do that? Because they're too dissimilar. That's a great example of heterogeneity in practice that we never talk about. But you can appreciate then that if I go down to human studies and say, well, are there also differences there? Well, some men are pigs and all that. But so there are differences in studies within human studies where you might say those differences are so great we shouldn't combine those studies. So we'll come back to this. This is what's called design heterogeneity. It's one of the 2 basic types of heterogeneity. And then finally, in meta-analysis, there's the assessment of publication bias. The way it's being done today is rather shallow, I would say. We have some ideas about how we might improve on that, and we'll get to that. So those are the sort of basics, and if you want to make this the sort of one slide that you remember and you say, well, what does meta-analysis do? It does these things. It provides a more precise estimate, which we talked about before, of the individual estimates from those studies. It combines those and pushes that confidence interval, makes it more narrow. Similarly, if those studies have dose-response effects within the study, you can do exactly the same thing for dose-response. So if I've got 5 studies, each of which has looked at a dose-response curve, I can combine those and get a single dose-response curve using a meta-analysis method. So that does the same sort of thing. It gives me the overall dose-response based on those 5 studies. Similarly, with this weighting process that we talked about before. The third point is actually fairly important because this is the only place where meta-analysis connects to that criteria-based method of causation, because what meta-analysis does for you is it evaluates consistency. Very simply put, one of the fundamental problems you have when you're evaluating a body of evidence is, so are these results consistent or not? Very important. I mean, if you look at the way people practice causal inference today, you would say that that's the single most important consideration is consistency. You see it all the time. You see people saying, well, a data is not consistent, therefore I don't know what to say about it. What meta-analysis does for you is provide you with an assessment of consistency. How do you do that? Well, it's pretty straightforward, actually. If the studies are similar enough that they can be combined, and I combine them into one value, by definition, they're consistent. We'll come back to that. The heterogeneity issue and publication bias, we've talked a little bit about. We'll get into that in a little more detail in a minute. So let's talk about these two issues, the first two here. Well, they're not the first two, actually the first and third, but we'll talk about those. And this is, again, going back to that same picture I showed you before. And this is just, and I don't mind repeating myself. Do you mind hearing me say a couple things twice? No? Okay. So we have the summary relative risk, a more precise estimate of that, and we have an assessment of consistency. Here's a good example. When I was, I'll tell you this story. A few years ago, I went to see my physician because I wasn't feeling well, and he said, oh, you have hypertension, which is something that runs in my family, and so I got treated for that. And then he said, oh, by the way, you have a nodule on your thyroid. I said, oh, that's good to know, I guess. He said, well, it's probably not a problem. So I had that taken off, and it was benign. That's good. But I couldn't talk for a while, which is not good. And I had to give a talk, like this one. So I gave the talk and didn't say a word. It was really kind of fun. I just pointed to stuff, just like I just did then. Yeah, it worked out just fine. Sorry. Okay. Let's go on. So I wanted to point this out, because remember, I was telling you about how meta-analysis fits into this broader notion of the criteria-based methods of causality, and these are a set of methods that have been around. In fact, this is the 50th year anniversary of the Hill criteria, this was written about in 1965, actually, and this is a picture of Austin Bradford Hill, who happened to be also the designer of the first randomized clinical trial, by the way, a very bright medical statistician. And he talks about these characteristics of an association, of evidence really, and how they are important to a causal inference, and consistency is a big deal there, and I just wanted to point that out because, as you can see, here's another example of a meta-analysis, and this is an example that you guys would probably be even more interested in, because this is about sugar-sweetened beverages and type 2 diabetes. And once again, you have this capacity to identify and to summarize across a variety of studies all of these different rate ratios, each of which has their own confidence limit, and you get this summary relative risk, this 1.16, you can see it down there, probably have a pointer here, but yes, right there, and the confidence interval around it, which is smaller than any of the other ones, as we've talked about before. And then this is, and we're going to talk about heterogeneity in a minute, the I-squared, which is a way to measure heterogeneity, is 40, and that's pretty good. 50 is sort of the number at which you're sort of concerned that there may be too much heterogeneity, but bottom line here, there's also a p-value, but we'll just stick with the I-squared. So this is another example. These are consistent results. Now you might look at those and say, well, gosh, they don't look very consistent to me, but in fact, given that you have a sort of quantitative way to summarize this evidence, you have a measure of how different they are statistically speaking, you can say that for all practical purposes, these are consistent. This is consistent evidence. And this is another example. This is actually a low-calorie sweetened beverages and type 2 diabetes meta-analysis. These two meta-analyses are just being finished up and will be submitted for publication soon. And this is exactly the same kind of process that we talked about before. So we have a variety of studies, and these are the crude values up here, and then we have the fully adjusted models down here. And in fact, we get this nice relative risk and the upper and lower limits of the confidence interval. Okay, so far so good. We'll keep going. Let's talk about heterogeneity, because that is an area, when I was talking before about heterogeneity, and we've talked a little bit about it, there are two types of heterogeneity, two basic types. We have design heterogeneity and statistical heterogeneity. For those of you who do meta-analysis and spend a lot of time thinking about meta-analysis, or you look at meta-analysis, pretty much The only type of heterogeneity you're going to see in a publication is this bottom one, this statistical heterogeneity. And what that does for you, you can read that. I hate reading slides for people, but I'll do it. It's a mathematical assessment of the variance. That sounds very too complicated. Fundamentally, statistical heterogeneity is a way to measure how overlapping the confidence intervals are. And if they're not, if they're overlapping, pretty much overlapping, the heterogeneity is going to be very low. If they're not overlapping, heterogeneity is going to be high. Okay, make sense? Okay, so that's statistical heterogeneity. There are a number of ways to assess it. We've looked at this before. There are two fundamental, at least two ways. There's a p-value there, which if it were 0.05 or smaller, that would mean that there was statistical heterogeneity, or the test would be positive. And the I-squared, which we talked about before. And the I-squared goes from 0 to 100. If you get to 50, you're, as I mentioned before, you are too high in heterogeneity. Now, what actually happens in practice, it's not like people say, oh my gosh, I've got an I-squared of 87. I've seen these. We shouldn't publish this. We should send this paper back. No, that's not what happens. It gets published anyway with the sort of caveat at the end that says something like, but we should be concerned about these results, something like that. So just to let you know, even though you might argue that you should not have done this meta-analysis in the first place because heterogeneity is too high, in practice, there's only a warning given, and no one says, oh, well, let's not do it in the first place. That's the way it goes. The second, or the first type, and this is the type where there's really been very little written about this. It makes perfect sense that you shouldn't put studies together if they're too different, but how to do that is now sort of, how shall I say, people really don't know how to do that, not in a formal way. So for example, when meta-analysis first showed up in the 1980s, folks would not put case control results and cohort studies together in the same meta-analysis because they would say, oh, no, after all, these are cohort studies, and that's different than a case control study. Today, that has gone away. And now it says, well, after all, the case control study is estimating the relative risk of a cohort study, so why not put them together? So that part of design heterogeneity has sort of disappeared. Nevertheless, there are issues here in design heterogeneity that would be reasonable to think about. What population is being studied? For example, if one study is only in men and the other study is only in women, does it make sense to put them together? From an exposure perspective, we'll do a sugar-sweetened beverages example here. If one study is measuring sugar-sweetened beverages defined as soda, period, versus another study that defines exposure to sugar-sweetened beverages as soda or pop, plus fruit juice, that could say, well, those are really different things, and I would suggest we not put those together. But as I pointed out, in practice today, there's no formal way to do this. What we've decided to do is to lay out the study design sufficiently. We do this in something called evidence mapping, which you may have heard about, in order so that you can at least make those kinds of decisions if you want to. There's also an issue here of which confounders. You know, confounders are sort of the bane of the existence of the epidemiologist. Which confounders were measured and controlled for in a given study? If one study is controlled for 6 confounders and the other is only controlled for 2, you might say those are very different studies and a rationale for not putting them together. How are we doing here? Oh, we're good. Now, I wanted to point this out. This is, you can't read this. This is good. This is a classic article on what are the components of a good meta-analysis, and you will see that right now I'm only talking about this assessment of heterogeneity, and we're going to talk about publication bias somewhere. It's in here somewhere. Those are just 2 issues in a whole variety of issues that apply to meta-analysis. So if you want to do a meta-analysis and you want to assess the quality of that meta-analysis, this would be a good reference for that. It's a Stroop paper in JAMA in 2000. And you can see it goes through a lot of things that you think would be appropriate. Do they define the problem? What's their hypothesis? The description of the study outcomes, pretty classic. How they searched for the literature. how they reported their methods, and then there's this assessment of heterogeneity. You notice this is all about statistical heterogeneity, and I'm pretty sure they say publication bias. Yeah, publication bias is right there. And you know the old disclosure of funding source issue as well. And what we've really been thinking about a lot lately is this assessment of heterogeneity, primarily design heterogeneity, and the publication bias problem. So let's talk about those a little bit more. How do you go about improving this situation of design heterogeneity, which we've talked about? We don't have a snappy answer to this. We only have a way to identify the characteristics of each study. So you might, in fact, if you look at most published meta-analyses, they don't give you very much information about the studies themselves, not to the level at which you'd like to have to be able to say how different are these studies. A way to do that is to do something called evidence mapping, which is really just one heck of a large table that lays out all the details of the studies so you can make these kinds of decisions. And then you determine the extent to which the studies differ by design, and you combine those studies that are sufficiently similar. It's as simple as that, if you think of that as simple. Now let's talk about publication bias, which is the tendency for studies with positive rather than negative or null results to be published. That's usually the way it's presented. You could argue that it could go the other way as well. It's the idea that maybe the process of publication itself is promoting one result over another. Now, the way you assess that in meta-analysis, and you've probably seen these pictures, they're kind of, I don't know, I'm a little, like I said, it's a little shallow. It's basically a statistical test. So basically you're saying, and these little dots that you see here are the results of each study. And fundamentally, the idea is there's a test to see whether the results in this triangle are pretty much the same as the results in this triangle, or are they different, and if they're, and in this particular picture, they're not. This is an example where publication bias doesn't exist, and this is an example here where publication bias does exist, and it really has to do with these two outliers that are sitting over there on the right-hand side of that right-hand triangle. Now, I don't think it takes very much thinking to say to yourself, well, why would I expect this to be a process that the publication of some observation of some biological phenomenon would necessarily be randomly distributed or not around some center line? I mean, for me, that says, well, that's good, but it doesn't seem to get to the heart of what publication bias is all about. So what we've done is to think about it in a very different way, and that is to say, so publication bias is about not just about what studies have been published, but what studies haven't been published. Then why not identify the studies that may have information on this same hypothesis but have not yet been published? And in dietary and nutritional epidemiology, nutritional factors, this is huge. For example, if you look at sugar-sweetened beverages or the low-calorie sweetened beverages literature, there are at least 15 major cohorts out there that very likely have information on sugar-sweetened beverages and, say, type 2 diabetes, but they've never been published. Now, I'm not saying whether they would be positive or negative. I'm simply saying that those studies, because they haven't been published, that to me is publication, the potential for publication bias. I can't tell you if it's biased or not. I can tell you there's information out there that hasn't appeared yet, but could appear, and would probably, or maybe not, make a huge difference in our assessment of this particular relationship. That seems to me to be a much better way, or not a better way, another way to assess this issue of the extent to which the results are biased or not. So we've talked about that in some of our new work. So let's, in the time we have remaining, let's just sort of go through the sugar-sweetened beverages and type 2 diabetes meta-analysis that we're submitting very soon. Just to let you know, There have been 2 prior meta-analyses on this subject, both of which, as you can see here, have high heterogeneity, and they reveal weak associations and some evidence of a dose-response relationship. And since the last one was published, there's new information. And that's probably, quite frankly, one of the best reasons for doing a new meta-analysis is because new studies come out. So the explanation for the association remains unclear, so we're going to do our own sugar-sweetened beverages and type 2 diabetes meta-analysis. These are our methods. They're pretty classic. You can see that we've done our searches. We're going to combine the data using random effects models. We didn't really talk about that today. They get into the statistical models. We evaluated the confounding. That is, we evaluated the extent to which each study measured confounders a priori. We calculated the summary relative risk. We assessed dose response and assessed publication bias qualitatively and quantitatively. And heterogeneity is not up there, but of course we did that too. And I showed you these results before, but here they are now. These are the studies that have looked at sugar-sweetened beverages and type 2 diabetes. This is what's called the extreme quantile comparison. So that's looking at those people who drank the most sugar-sweetened beverages versus those who drank the least amount. And so, like I said, we've seen this before. So this is the summary relative risk for the studies of 1.16, and these are the confidence intervals. Now, by any one stretch of the imagination, this is an extraordinarily weak association. What does that mean? That means that it would not take very much for any unmeasured confounder, these are unmeasured factors in the studies, to push that 1.16 back to 1. We did find that there was not a significant amount of heterogeneity, so these are consistent results, as we talked about before. This is our dose-response relationship, and you can see it's kind of a peculiar relationship where we actually, and this is one of the differences between our study and the most recent meta-analysis, is that rather than converting the servings into actual quantitative amounts, which involves a variety of assumptions, we simply use the information that was reported by the participants in their studies. How many servings a week of sugar-sweetened beverages? And if you do that, you can see here that what you get is an I-squared of 0, which means there's no heterogeneity at all, and a very peculiar relationship that you could call a dose-response relationship, except that when you get up to the highest levels, it drops down here to 1.04. So it's kind of well, I would say that's not really a very good dose-response relationship. That's what I would say. And as it says down here, each row represents a meta-analysis of relative risks. So each one of these is the meta-relative risk across those studies that reported dose response. And so what we concluded from this is that the relationship are more homogeneous than previous estimates. That's a good thing. The relationship is extraordinarily weak, and it lacks dose response. And whether the associations will be reduced further from confounding remains unclear. And then there are these 15 studies that we know exist that we don't have information on, which would actually be very good to have that information if we could get it. Let me acknowledge my collaborators here, Dr. Alexander, Dr. Altice, Dr. Frankenfeld, and the support for this came from the Coca-Cola Company with all of the conditions there. So fundamentally, meta-analysis, very powerful method that has some important strengths. It allows you to combine information across studies. That's huge. It allows you to assess the extent to which those studies are different, statistically different. That's the statistical heterogeneity factor. It also permits you to make more precise estimates of a dose-response relationship. That's a good thing. What it does not do for you, and you'll see people who make this claim from observational meta-analyses, is allow you to make a causal claim simply on the basis of a meta-analysis. And why is that? Because all it does is it assesses consistency. That's the only thing it does for you. Yes, it provides you with a relative risk, but you still have to decide whether that relative risk is big or little and how that fits into a causal assessment. Yes, it gives you a more precise estimate of the dose-response curve, but you still have to decide what does that dose-response curve look like. So from a causation perspective, it answers the question of consistency. That's very important, but that's all it does. So be very cautious if you see a meta-analysis of observational data and you see someone saying, well, there we know the answer. No, no, no. You know a part of the answer. You know this overall summary estimate, and you know whether the results are consistent or not. And that's all you know. And I'm going to stop, and thank you. I'll take questions.